Numerous neuroimaging studies have been conducted in an attempt to explain the physiological mechanisms of OCD. A 2008 study by Huey and others begins by acknowledging that the orbitofrontal cortex and head of the caudate were the only brain areas that significantly and consistently showed increased tracer uptake for tomography tests in OCD patients. A more recent 2014 report by Gaikwad furthers this by indicating the results of neuroimaging studies in OCD patients have most consistently involved the orbitofrontal cortex, the cingulate cortex, and the basal ganglia, and regions within the parietal lobe. This is all in relation to the pathophysiology of obsessions and compulsions. This suggests that OCD is factorial and that it exists in different forms. Gaikwad's same 2014 report continues to illustrate how the dysregulation of the serotonergic system has been theorized mainly because of the effectiveness of selective serotonin reuptake inhibitors in improving obsessions and compulsions. These benefits also led to finding that deviations of the dopaminergic system have been linked to OCD. The report continues to identify evidence that indicates the involvement of the glutamatergic system in OCD. Some evidence suggesting the involvement of nitric oxide in OCD was even found according to a 2005 study by Atmaker and others. This study found that OCD patients have higher nitric oxide levels in their plasma compared to those without OCD, and that these levels positively correlated with the severity of OCD symptoms. Despite the advances in psychopharmacology and psychotherapeutic treatments, a 2014 paper by Brunelin and others claims that more than 40% of patients with OCD do not respond to these conventional treatment approaches. Transcranial direct current stimulation has been recently studied as a therapeutic tool to help treat patients with resistant OCD symptoms. A report conducted by Nietzsche 2015 has found that TDCS's primary mechanism of action is a polarity-dependent subthreshold shift of resting membrane potentials. The after effects of stimulation were shown to depend on the glutamatergic system. Beyond these effects, Nietzsche also found that TDCS has been shown to alter cortical as well as cortico-subcortical functional network connectivity. Nietzsche 2011 supported these theories with a study which examined the evidence for the physiological mechanisms of TDCS and mainly evidence that TDCS modulates synaptic strength within the motor cortex. The authors were able to analyse animal studies which recognised that polarisation of the cortex is capable of modulating cortical excitability and neuronal firing rates. Also that these effects can last numerous hours after the stimulation period. They also found that human studies suggest TDCS modulates cortical excitability during stimulation by non-synaptic changes of the cells, but acknowledged evidence that the after effects of TDCS are driven by synaptic modification. These findings suggest that TDCS does modulate synaptic strength within the cortex, and it is said that the evidence points to the involvement of intracortical neurons. Most effective treatments for OCD are pharmacological treatments using serotonin reuptake inhibitors, behavioural treatment, and using the response exposure and prevention technique according to Gaikwad 2014. Another 2014 study by Brunelin and others ran an analysis on 77 patients with OCD who received active TDCS with different electrode settings. Most of these studies reported a significant effect of TDCS on OCD symptoms. Many of the studies also reported beneficial effects from TDCS on other symptoms that are often observed in patients with OCD, such as depression and anxiety. This same 2014 study examined numerous other neurophysiological studies and have reported that depending on the electrode polarity and current intensity, TDCS may increase cortical excitability in the vicinity of the node, whereas cathodal TDCS may decrease it. The study notes the effects of TDCS are not restricted to the area beneath the electrodes and could reach a widespread network of cortical and subcortical regions that are not connected to the targeted region. The ability of TDCS to modulate a network is of particular interest since abnormal activity and connectivity within the previous networks discussed is described in patients with OCD. It was deduced that applying TDCS to the specific areas of the brain affected in OCD yielded the best relief. Many of the problems that need to be addressed for future research have also been outlined in Brunelin's 2014 study. In his review of 12 other studies, none of them were sham-controlled. Most of them were case reports, and only 2 studies included more than 10 patients, limiting the interpretation of results due to the small sample size. The number and duration of TDCS sessions was overly varied and more tailored to depression. Most of the patients included in the reviewed studies even had different clinical characteristics and were treated with different medication which may influence TDCS effects. And finally, Very few studies have investigated the brain correlations of symptom improvement after TDCS in OCD patients. Investigating biological effects of TDCS in patients with OCD will provide a better understanding of the physiological psychology of OCD and of TDCS's mechanisms of action.